Today I will be talking about knowledge graphs, and the question is, what are knowledge graphs, right? First, some backgrounds, right? Of course, all of you remember in the year 2000 there was this proclamation that by year 2010 we will know the function of most of the genes in Arabidopsis, but a quarter of a century forward, we only know the function of around 12% of genes. And by this, I mean, we know the biological process they're involved in, the cellular components that they're found in the molecular function, right? So only 12% of genes have been experimentally verified. What's scary is that the number of articles reporting gene function has peaked in 2008, and the number of new genes with characterized functions have been dropping ever since. This is surprising because the number of papers containing Arabidopsis keyword has peaked in 2010 and remained constant, right? So it looks like that for some reason the discovery of new gene functions has dropped, but the number of papers has not. So how could we fix this? So I believe strongly that one way to fix this is to make very accurate, very good predictions about what the different genes are doing, which can then guide experimentalists in their work, right? I mean, very often genes do not have phenotype, but maybe they don't have phenotypes because we look at the wrong time and in the wrong place under the wrong conditions. But again, the problem there is that actually the performance of the gene function prediction methods are not that good, and they have not improved a lot in the recent years. I mean, this is a conclusion from 6 years ago from the CAFA challenge, where they say, well, okay, there have been some improvement, but it's actually very minor, right? And actually, also looking at the recent literature, there have not been any real breakthroughs in this. So it looks like we are somehow at the standstill. So what do we need to predict gene function? So we need 3 ingredients. The first ingredient is the omics data. So this could be, for example, gene expression in form of RNA sequencing. This could be protein sequences, genome sequences, and so on and so on. And we use large-scale studies. So, for example, the gene expression atlases, where if you have thousands of samples for Arabidopsis. The second ingredient that you need is the gold standard. So it's basically the collection of genes which somebody has experimentally characterized. This is like the ground truth, right? So this information is found in the scientific literature. So somebody has to read those papers, extract the information, and put it in a way that the computer can read. And the third ingredient is a gene function prediction method. So this could be, for example, methods such as sequence similarity approaches, co-expression, and so on and so on. So the function prediction method uses the omics data and the gold standard to, for example, build, let's say, co-expression networks where we know what the green genes are doing, and then we can predict what the gray genes are doing because they're connected to it. So how can we improve gene prediction? Well, we can improve it by improving those 3 different ingredients. So for the omics data, we could have, new data and new types of data. For example, single-cell sequencing is relatively new data, and more of it would allow us to build stronger models. We could also use more gold standard data. So the more we know what the genes are doing, the better we can actually make new predictions, right? And the third ingredient, well, we could use more clever, more powerful gene function prediction methods to squeeze more information out of the omics data and the gold standard. So today, I would like to show you a new approach that we developed in the lab that kind of addresses all 3 different domains. So I'll show you a new way of getting new data, getting more gold standard data, and also I'll propose methods to make new predictions. So the disclaimer is kind of, okay? So it's, I'm cheating here a little bit, but I'll tell you why it's still very valid. So it was a boring weekend in 2023, and I think like all of you, I was playing with ChatGPT, and I looked at an abstract. So this is from Stefan Person's group. So I was doing my PhD there, and when you look at an abstract, you can see this abstract is actually packed with information about gene function, right? So the abstracts typically tell you the main findings. And the abstracts here tell you what different proteins are doing and how they are related to each other. So the idea was here, can we take this abstract and use ChatGPT to extract gene functional information from it? And this is the abstract that the prompt that I used. I told ChatGPT, okay, write a very short summary about the function of genes in this abstract and give me the information such as gene affects process, gene localizes to X, or gene interacts with Y. And here I paste in the abstract, and because ChatGPT is forgetful, I yelled at it, saying again, give me those things in this particular format. And indeed, it listened, right? So it was able to identify pairs of a relationship. So for example, cellulose synthesized at plasma membrane, cortical microtubules guide cellulose synthesis, and so on and so on. So knowing that this worked, we then analyzed all of the papers that contain an Arabidopsis gene in their abstract, which was around 71,000 abstracts. So for the journals in red, we could analyze abstracts, and for the ones in blue, we could also get full text papers. So we analyzed those papers using the prompts that I showed you. So it took around 7 days and cost around $3,000. And from this, we got a knowledge graph. So realize that here we have always two entities. Let's say a protein and another protein, and they are connected by some relationship, right? So you can actually represent this as a huge network, right? So this is the knowledge graph where the entities can be genes. They can be also phenotypes, organs, metabolites, chemicals, basically whatever. And they are connected by edges showing the relationship. So for example, here we see that PSAD1-1 mutant downregulates mRNA expression of genes involved in photosynthesis. While in this mutant was analyzed for phosphorylation of thylakoid proteins in some paper. So in total, we extracted 2 million entities, and we found around 5 million edges between those entities. So how accurate is actually a large language models in extracting information? So we did find some errors. So for example, here you can see an example of a sentence from this publication. So the large language models correctly said that SNPs change binding of transcription factors, right? But here it correctly said that the SNP is a genomic variation. But here binding of transcription factors is said it's a gene. It's not a gene, right? It's more like a process and that the SNP participate. So overall, we found that the extracted information is of quite high accuracy. So for predicting the types of the entities, we observed for most cases accuracy of, let's say, around 85%. And also the relationships between the entities was more than 80%. So typically the errors that the large language models do is that they confuse a fact with like a future goal. So for example, in a paper, somebody could say, in the future, we want to test if protein A and B interact. And the large language models will say protein A and B interact, right? So this is something that we're working with. So What are the most common entities among the 2 million entities that we found? Well, the most common one is Arabidopsis thaliana, because we were looking for Arabidopsis gene IDs, but we also see entities such as salt stress, drought, auxin, cold, and others. The most common relationships are head, show, contain, and regulate and involve in. And the most common entity types is gene, which makes sense because we're looking for abstracts which contain Arabidopsis gene names. Then we have phenotype, because very often papers report phenotypes of mutants. Then organism, metabolite, and so on and so on. So for each of the different entity types, so for example, for gene, the most common entity was just genes, because people just write those genes show this phenotype. But we also see something more specific as HY5, YB, PIF4, and so on and so on. For phenotypes, well, plant growth is the most reported phenotype, flowering, root growth, and expression. For organisms, not surprisingly, Arabidopsis, but we also find rice, maize, and others. For metabolites, auxin is apparently a big thing, ethylene, reactive oxygen species, and so on. And yes, so this is just like a description of the knowledge graph. So let's take a look what else we could get. So we're very happy to see that we were able to identify protein-protein interactions from text. So here you can, for example, see interaction network of SESA1. So this is a gene involved in cellulose biosynthesis. And here you can see the different genes that have been documented that this gene interacts with, isolated from literature. So let me just show you quickly how this works, right? So this is the database that we developed. So this is the entity page for cellulose synthase 1. And for example, if we want to look at genes only, we can select genes here and then interacts with. So this will only report, this will only show genes and interactions of this gene. And what's, I think, quite interesting is you can click on an edge of this given gene, and this will show you also what the researchers did to actually prove this interaction, right? So we made a large language model read this paper and find all of the things that the researchers did to prove it. So in this case, you can see that they did these two hybrids and BIF as, right? What's also possible is you can also ask the large language model to validate this. So as I showed you, it can make mistakes sometimes. So when you click on this link, what will happen is our tool will take this text, it will then send it to large language model, and it will ask, is this really true? And in here, you can see that, that this is the sentence that underpins this particular edge. And overall, the large language model says, okay, this is actually a correct, edge. So you can basically test the validity of those edges real time using our tool. Yes, but what was very cool here is that all in all, we identified 11,000 protein-protein interactions, and 8,200 were found specifically by our tools. So on the left here, you see BioGrid, which is like the gold standard repository of protein-protein interactions available. And I would argue that actually our interactions are probably better because BioGrid contains things such as just, the outputs from yeast to hybrid, while our interactions are based on somebody actually writing that protein A interacts with protein B. So I would argue that our interactions are probably more solid. For another example, so here we have MIP46, right? So those of you working on secondary cell will know those genes. So here we have genes regulating MYP46 and then the targets of MYP46. And when we compared our gene regulatory network with the gold standard, which is AGRIS, you could see that we identified almost 50,000 new edges, which is more than doubling of the available gene regulatory network. Yes, so I will show you some other functionalities of our database. So as you could see here, we could validate the edges. Our tool provides you ways to ask many questions. So for example, it doesn't need to be gene-centric. You can actually look at relationships between any entities. So here, we're looking at the heat stress. And here you see a knowledge graph comprised of 3,200 nodes and almost 1,000 papers. And for example, if you're interested in knowing which metabolites change upon heat stress, you could go here and select chemical. And for example, if you want to know which metabolites are increased during heat stress, you could here, for example, select induces and increased, like this. And here you will see the different metabolites that were increased upon heat stress. So you see things such as gibberellins and so on. And again, you can validate the edge. And then you can see this particular edge. The large language model thinks it's incorrect because here it says that the synthesis of those metabolites was increased, but the large language model here is very picky, and it says, well, just because the synthesis is increased doesn't mean that the metabolite goes up. So it's a bit nitpicky here, I would say. Okay, so let me show you some more examples and some of the issues that are actually encountered. So one issue is that, for example, some entities are not resolved. So this is a protein interaction network of TOC75, which is involved in protein import into chloroplast. And you can see that there are some entities which are repeated. So for example, we have TOC80, TOC34, and TOC34. Right? So the problem here is that one paper says TOC34, another paper says 80 TOC34, right? And this sounds like a trivial issues, right? I mean, it's possible to solve for genes, but imagine we have 2 million of entities, and it's impossible to resolve it for all. Another example here is it's not just the entities that are a problem, it's also the edges. So for example, here we see different alliterations of the same edge type. For example, upregulates with a dash versus upregulates without a dash, right? Again, it's a silly problem, but remember we have 5 million edges, and it's impossible to actually solve this in a good way programmatically. Yes, so the conclusions from the talk here is that going back to those three objectives, more omics data. Yes, we have expanded available protein-protein interaction and gene regulatory data by simply excavating it from the literature. So far, this information was not accessible to bioinformaticians. This information was simply trapped in papers. And from the perspective of computational biologists, it was useless information. But by using an approach like this, you can actually liberate this data and expand the gold standard for bioinformaticians. So this is what I meant kind of, and I'm kind of cheating, right? Yes, also the gold standard data can be expanded. So for example, here we can connect genes and mutants to other entities, and those entities themselves can represent gene ontology terms. So for example, for example, photosynthetic electron flow has its own gene ontology term that we could then assign to this gene. And third, better function prediction methods. Well, such a knowledge graph can be actually analyzed to make new predictions, which I will describe in a minute. So what can we do in the future? So I think there are 5 steps that are quite exciting. So one thing is, until now we have been using existing large language models such as, for example, ChatGPT, but those models are huge, right? Those models know who the president of USA was in the 19th century. So this is not something that we need actually to make a knowledge graph like this. So what we're doing right now is we're using those highly performing but huge large language models to generate training data for smaller models. Right? So we basically want to use the output from highly performing models to train lightweight but customized and specialized large language models, which are just trained to make those knowledge graphs, or just trained to make some predictions. Yes, so the next thing is, yes, we want to analyze all of the plant literature, so not just abstracts containing Arabidopsis gene IDs, but the question is, how can we do it in a sustainable way, right? So one of the problems I showed you is that there are different alliterations of the same thing. So for example, we have here drought tolerance written by a high number of papers, but some papers also wrote tolerance to drought, which is the same thing, just alliterated, right? So there are many different ways of saying the same thing. And the way we want to solve it is we want to actually feed back those entities to the large language model and ask it, well, what do you think? Which of those entities are actually the same thing? And we can actually use it to put the same entities into the same cluster. And it actually works pretty well. I just did not have time to show you about that. And third, yes, we can actually mine this knowledge graph, right, to identify the gene ontology terms. So, for example, this one here is clearly photosynthesis. This one is thylakoids, subcellular localization. This one here could be, for example, photosystem, right? But again, the question is, how can we do that? And here the solution, again, I believe, are large language models, right? I mean, we can provide large language models with definitions for the gene ontologies. We can then provide a large language model with an entity and its description, and then ask it, well, which GO do you think this entity represents, right? And we could do it in a high throughput fashion. Fourth, what we want to do is we want to generate something like ChatGPT, but for plant literature, right? So, you know, ChatGPT is great, but it's also one of the most confident bullshitters, right? So if you ask it something, it will say, yes, it's like this, and this is PubMed ID, and then it just gives you some random number. What we want to do is we actually want to build a tool where the user asks a question, right? Then this question can be used to identify entities in the knowledge graph that are connected to this question. Then the large language model should take the identified parts of the knowledge graph and then generate texts, right? And this is something that we also have going. So if you go to our tool, and I'm going to use Dolph here as an example. So if we go to the author page of DOLF, you can see there are 68 papers that we could analyze for DOLF. So this is the knowledge graph of DOLF. And here we could say, okay, we're only going to look at the genes. And here we could say, okay, I'm going to give you this knowledge graph, which is summarized as just as texts here. And then we can ask, okay, generate the summary of captured by this knowledge graph, and also provide us with the references, right? So the advantage here is that this text is only based on the knowledge graph. We told the large language model explicitly do not use your existing knowledge, right? And by doing this, we can avoid hallucinations. And then the fifth, well, when you look at those knowledge graphs, you start to think, right? I mean, and realize that there are some relationships, right? So, for example, in this example, we have gene A interacts with gene B interacts with gene A, and then gene A or its protein localizes to chloroplast stroma. And then you could ask, I mean, you could make a hypothesis. Well, does gene B localize to chloroplast stroma, right? So I believe you could actually mine this knowledge graph and also build customized large language models which are trained for this kind of a reasoning that we are doing to actually identify new links between entities, which could then serve as hypotheses, and also maybe identify new entities to begin with. And this could be done by using large language models. I mean, they're quite good at reasoning, especially if you give them some examples. But we could also use some kind of a symbolic reasoning, which is based on well-defined rules or even probabilistic reasoning. Yes, so this is the end of my talk. So I would like to acknowledge my group. So this is my group from Singapore, as you can see. And the people working on this projects are Shan-Chun, Ken, and Manoj. And I would like to thank you for your attention and take any questions.